Hi everyone, welcome to another session for my science journey, but this time around we are hosting a very important conversation, a conversation around one of the genetic diseases that is very prevalent in the African populations, but we have a lot of stigma and misinformation that circulates around it. And today, we are just hoping that with this conversation, as well as with efforts that are continuing to take place around these conversations around the sickle cell disease, we are more enlightened and we will be able to enlighten our communities as well. And, we hope that if this is your first time to hear about an in-depth conversation about sickle cell, that it doesn't stop with you and that this conversation keeps going on, from you to your communities and to the communities around you. So thank you so much for joining today. We are privileged to be hosting an incredible group of facilitators that are experts in this field and people that have worked around, the sickle cell disease in the African context. And so we'll be tapping into their wealth of wisdom, wealth of knowledge, and we are so glad to have them on board. So before we start, I'd like to quickly introduce our facilitators. If you could please unmute yourself and just say hello, who you are, as well as the sort of like role you play. Especially around the sickle cell disease conversation, we would really appreciate that. So I think I would start with Mohammed. Mohammed, if you can hear me, please go
Thank you
ahead.
so much, Ruth. Hello, everyone. This is Dr. Mohammed Zahir. I am a molecular geneticist and a genetic counseling practitioner. I'm based in Dar es Salaam, Tanzania, at Muhimbili University, and I'm also the General Secretary of the Tanzania Human Genetics Organization. Thank you very much, Ruth, for doing this. Congratulations to my science journey, and we look forward to these very important discussions as we commemorate the Sickle Cell Day.
Thank you so much. And thank you for also highlighting that. I think if you are not aware that tomorrow is World Sickle Cell Day, and so this workshop just comes a day before that, which is a great thing. And hopefully with the information you have now, tomorrow you can play around and spark the conversations around sickle cell disease. Thank you so much, Mohammed. Siana?
Hi everyone. Ruth, thank you so much for organizing this important session. As you say, sickle cell disease is a disease that we need to be talking about in Africa. And June is Sickle Cell Disease Awareness Month. So it's really a good timing that we are doing this today. And I'm really hoping that we will all learn from one another and take the agenda of fighting sickle cell disease forward in Africa. My name is Siana Nkia. I'm a senior lecturer in the Department of Biochemistry and Molecular Biology at Muhimbili University. I've worked with the sickle cell program in Tanzania for more than 15 years. And as part of that, I've participated in different roles within the sickle cell program of Tanzania and within the Africa region to conduct research for sickle cell disease, as well as participating in different initiatives. I'm really happy to be here today to be contributing toward this journey. Thank you so much.
Thank you so much, Dr. Siana. And now we'll have Arafa.
Yes, good morning, everybody. My name is Arafa Salim Said. I'm a sickle cell advocate, but a genetic counselor, and I'm the founder of Sickle Cell Disease Patients Community of Tanzania. Thank you so much for having me here. Thank you so much.
Thank you, Arafa. Sainabu?
Good morning, everyone. My name is Sena Bolandu, and I am currently a PhD researcher in human genetics in the Queen Mary University of London. I am also the founder of the Human Genetics Awareness Association, and I am pleased to be here today. Thank you, Ruth, for having us, especially to talk about human genetics in the context of sickle cell disease.
Thank you, Sainabu. And finally, Helen?
Good morning, everyone. My name is Helen Shikanda. I am a health and science journalist and I work for the Nation Media Group in Nairobi. I'm so glad to be here because I'll be learning a lot. And as a journalist, I know we like such things, especially when the awareness days are coming up. So I know I'll even be getting a story idea to work on. And later today, I'll be moderating one of the sessions, and I'm happy to be here. Looking forward to the sessions that we'll have today. Thank you so much, Ruth.
Thank you, Helen. And finally, I'd like to reintroduce my co-facilitator, Lena.
Good morning, everyone. First of all, thank you so much for joining. My name is Lena Ochieng. I'm a PhD student at the University of Oxford, and I'm so glad to be doing this together with Ruth.
All right. Sorry, guys. Yeah, so that's everyone. And thank you so much, everyone, for, being on board and doing it with us. And we are hoping that every one of us that is here to learn will be able to pick a thing or two. And hopefully it will be beneficial, not just for you, your family members, but also for your community. And now I'll just go quickly. Yeah, so sorry, I'm just being reminded to record the session and to also start the translation, so I will quickly do that. But before we start, let me quickly share the agenda of the day, so as to give you a brief overview of, the conversation. Hi, everyone. Welcome to another session for my science. Sorry about that. So this is the agenda for the day. As you can see, we've started a little bit behind time, but we'll try to catch up. So we will have a brief introduction to genetics and genetic conditions, just to give people an overview of when we say genetic diseases, what do we mean, before we sort of like come down to sickle cell disease. And, our experts in the house, Dr. Mohammed and Dr. Siana, will be able to introduce this. Then we'll quickly jump into two panel discussions around sickle cell disease and have a group photo. Then after the group photo, we're gonna go into the breakout rooms. So just to mention that the people joining us on the live stream, you'll be able to join the conversations until the panel session. And once we do the group photo, you're free to leave, as the breakout rooms will not be live streamed directly to you. But hopefully what we are able to live stream to you is still as beneficial to you and your communities as well. So yeah, this is the rough agenda. You can quickly browse through it if you need to refer back from our workshop website at My Science Journey. Yeah, so if you just, type on Google or, whatever browser you're using, you should be able to see that. Okay, great. So let me quickly start the recording. But while I'm doing that, I'd like to invite Dr. Mohammed to start sharing his screen so that he could give us an introduction to genetics and genetic conditions. Thank you, Dr. Mohammed. We can see your slides. You are good to go.
Thank you so much, Ruth. Can you confirm if you can hear me?
Yes, we can hear you
Fantastic.
loud and clear.
All right. So good morning once again, everyone. And my job this morning for the next 10 minutes is basically to introduce the topic of genetics and genomics. Particularly, I'm told that this particular audience is not a scientist audience. And so it is very important that as we conduct our advocacy, that we introduce the topic and the subject in a way that is palatable and understandable to all, not just the scientists. So I will take the next 10 minutes and hopefully give you a bit of an insight around what is genetics and what is genetic conditions. And that hopefully will give you an understanding of what it is that we have inside our cells as instructions, and that will make us behave in a certain way or make us who we really are. So each of us in this world obviously is a product of their father and their mother. And one of the things that we have to understand is that our body is made up of trillions of cells. And for majority, apart from red blood cells, majority of the cells have something called the nucleus. Think of it as the engine of a car. Now, that engine of a car has to function properly for the cell in the body to be able to produce and function in a way that the body will be able to work. And in this particular cell, we've got something called the DNA. And this particular DNA has 46 chromosomes. And to give you context, think of it this way. We've got a total of, if we imagine that each chromosome is like a shelf, if we go to a public library and we see different shelves. Now think of the nucleus or the DNA as your public library, where every shelf represents a particular chromosome. And so in human beings, for each nucleated cell, we have a total of 46 chromosomes or 46 shells. And these are actually 23 pairs. Now, when we say what is a gene? Now, if we zoom into one particular shelf and we pull out one particular book, then that particular book will represent a gene. And so each book will have its own instruction for a specific function. And that's why when we say, if I'm going to look for a biology book, I will look for a specific biology-based shelf, and then I will try and see which specific book I want, and that book will represent the gene, which will give you the instruction that you would want to hear. And so, to give you context, this is how the basic structure of DNA looks like in human beings. Now, what is very important is for us to understand that we are a product of our parents, meaning that of these 46 shells, we get 23 from our mother and 23 from our father. That means a total, we have 46 chromosomes. And it is very important to also understand that every child is a unique combination of these books or genes from both parents. And that is why many times you will also realize that children can actually look like both their mom and dad and still be their own person. Now, you might also be asking yourself is why is it that sometimes we look alike? And this is because we share genes that influence our traits and health conditions. If we look at a 3-generation family, for example, when you have the mother, the grandmother, and the daughter, you realize that sometimes there are certain traits, physical traits, that are passed on, like maybe the hair type or even the eye color or the skin color. Sometimes you say, oh, this smile, look, she smiles completely like her mother, or even the blood group, or even the height. And so this is the reason why we start to really look alike within families, because in fact, genes are one of the reasons that are shared, which are shared from parents to offspring, and the reason why we've got similar characteristics and some health conditions within the same family. So we may look different, but we are all connected through our genes. But at the same time, I also want to reiterate the fact that while we are, of course, receiving these 23 shelves from each parent and a combination of these books and genes from each parent, we are also very unique, because these small differences in the way that we get the books from our parents help make each of us very, very unique. And that means that these differences can come from either your skin color or your different hair type or ethnic groups, your height. All these things have an influence through the genetic inheritance. And of course, majority of these genetic differences are completely normal. And this is the reason why In fact, they are part of what makes us, every person, unique. But when we talk about a genetic condition, what really is a genetic condition? A genetic condition happens when sometimes there is a small change in this instruction book, in that gene which is supposed to give you instructions on how to work, that actually makes healthy proteins and help the body to work properly. Now, when there are changes in those instructions, that particular protein is not produced properly or insufficiently or probably too much. And so the body may not work properly the way it should. And that is what we define as a genetic condition. Now, not every change that happens in the instruction will cause disease, but there are some changes that can lead to genetic conditions. And what is very, very important as a take-home message for most of us here is that these genetic conditions are actually nobody's fault. We should not stereotype, stigmatize anyone to say that this is something that was caused by a particular parent or because of a particular reason. No, this is not a curse. This is not a punishment. It is not caused by bad parenting, and this is not contagious. All right? In fact, the fact is these genetic conditions are medical conditions, and no one chooses the genes that they inherit. Therefore, no one is to be blamed. And therefore, what we really need to be working towards is understanding one another to bring compassion. And through compassion, we will build stronger communities. So, why does the genetics matter so much? Is because when we are informed, when we have enough knowledge about our specific genetic problem, we are empowering our families. And by empowering our families, we will have better health options, we will improve our health options, and we will have better or stronger communities. Because understanding genetics and how they affect our bodies will help us understand how to prevent these kinds of problems, how to make informed decisions. We will also be breaking the stigma and breaking the stereotypes that are normally attached to these genetic conditions. And we will also be able to provide support to these families and communities with kindness. So very, very important is to understand why genetics matters, because with knowledge, we will also associate that to health. So we really should be using what we learn to build a healthier future for our children. And lastly, I just want to say that, to leave you with this message is that when we talk about from genes to our health, we have to understand that our genes, these are those books that we got from the shelves that make up the library. They carry instructions about and that tell us how our bodies work, including our blood. So we go from those genes or those books to producing proteins, and these proteins, are important for functioning of the body. And of course, these proteins are also involved in production of these blood cells. And I'm sure Dr. Siana will touch upon how these cells are so important, particularly in the context of sickle cell disease. So really, really important. Changes in genes can affect how these blood cells work. Understanding these genes and the instructions will help us understand conditions such as sickle cell disease. So with that, I want to thank you very, very much. And we will, of course, meet again each other during the panel discussion. Over to you, Ruth.
Thank you so much, Dr. Mohammed, for that very nice introduction. And I think I would give two people a quick opportunity to ask Dr. Mohammed quick questions if they have. So the first person to raise their hand or type in the chat box if there is any questions. Okay, I think silence means that we do not have questions, which is a good thing and also a bad thing, but okay. Let me lean on the good thing that it means that we've all clearly understood the introduction to genetic conditions. And so with that, I'd like to invite Dr. Siana to please share her screen and give us an introduction to sickle cell disease.
Hello again. I hope you can see my screen.
Yes, we can see your screen.
Okay. All right. So again, thank you so much, Ruth, for giving me this opportunity to talk today on a subject that is very close to my heart and one that I've been engaged for a number of years. And I should also say that I'm speaking also on behalf of so many people that we have been working together, including Dr. Mohammed and Arafa, who are part of facilitators for this event, for this session. So I've been given a task to talk about sickle cell disease, genotype testing, and blood donation. And I'm really glad that I'm speaking after Mohammed, because he has laid a very good foundation of the genetic disorders and how they come about. So we spoke about June being a sickle cell disease awareness month. And we also say that sickle cell disease is an important disease, especially for Africa, because it is highly prevalent. So we know that we have up to 500 babies that are born with sickle cell disease annually, and this is globally. But more than 80% of these babies, these individuals, reside in Africa. For example, in Tanzania alone, and this could be similar to many African countries, we have up to 15,000 babies born annually. So it's a highly prevalent condition. So we should really be talking about it more and more and having more people on the course to fight this disease. So sickle cell disease is a genetic disorder, and it's a disorder that affects hemoglobin. I'm sure many of us from our basic biology, we know a bit about hemoglobin molecule and the importance of hemoglobin. So I'm really glad that Dr. Mohammed touched about the different genes. So sickle cell disease itself is a defect that occurs due to a change in a gene that is responsible for synthesizing hemoglobin molecule. And we can see from this figure that the normal red blood cell is usually round and smooth. But when we have an abnormal hemoglobin resulting from the defect in the hemoglobin gene, we have a different shape of the red blood cell, which is now sickled and rough. And this is very important for us to understand, because this is actually the central hallmark of all the complications that we see in sickle cell disease, the way this red blood cell is shaped, but also the fact that the hemoglobin within this cell is an abnormal hemoglobin. So you will see that normally when you have normal red blood cells, and because we know red blood cells go throughout all the body, yeah, they travel in different blood vessels with different sizes, the big vessels as well as the smallest vessels. You see that they need to find a way of passing very smoothly. So usually the normal red blood cells would pass through the different blood vessels very smoothly. But that's not the case with the sickled cells. Of course, they are rough, and so they are sticky, and that is really the central way of how the different complications arise. So we know that hemoglobin as a molecule has a very critical, crucial role of transporting oxygen across the body. So you can imagine that if we have, so the normal hemoglobin is called, normally we call it adult hemoglobin. So when someone is born, they are born with fetal hemoglobin, which is one type of hemoglobin, but immediately that is transformed into adult hemoglobin, which is capable of carrying oxygen in environments at which an adult is expected to be living, which is different from the environment that the fetus lives. But then when we have someone with sickle cell disease, they carry the abnormal hemoglobin, which we call hemoglobin S. And the among key differences between the two hemoglobin molecules is that the hemoglobin S is not capable of performing its responsibility, its key responsibility of transporting oxygen thoroughly in the body, because its affinity is different. Its affinity to oxygen is different. So you see that because that fails, you see after the release of the oxygen, you have the sickled red blood cell. And again, from this, you start because the red blood cells that we have are damaged, their lifespan is also shortened. And the body senses that these cells are abnormal. So the body is also trying to get rid of them. So we have few of red blood cells, which leads to anemia. But also because of the red blood cells, which are sticky, we have blockage of blood vessels with the sickled cells, which leads to what we call vaso-occlusion. But because the liver is sensing that these red blood cells are not fit, they try to break this down. We have a lot of hemolysis, and we also have damage to the cell linings of blood vessels, which we call endothelial dysfunction. And because there's all of this happening, the immune system is also on alert. And so we have a lot of increased inflammation. And all of this, and because we are talking about red blood cells, they go through the different organs of the body. We also have organ damage to the different organs that we have. So how is sickle cell disease inherited? I'm really glad that Dr. Mohammed already covered the basics of genetics. So for sickle cell disease, we already saw that this is a defect that happens in the hemoglobin gene. And you can see here, it's just a single change. So from the books that Dr. Mohammed was showing, let's assume that this is book that we call hemoglobin gene. And in that book, in one of the chapters, we see that one of the letters has changed. Instead of having A, we have now T. And this just one change leads to us producing a mutant protein, is what we have seen. So the way this is inherited, and this is really important, this is a recessive condition, meaning that for you to inherit the disease, you need to inherit the mutant gene from both parents. This is very important knowledge because sometimes, and especially in Africa, we all know that bad babies, this is a mother's fault. So sickle cell disease inheritance is something that happens from both parents. You need to inherit the defected form of the gene from each parent. So you will see from this example that when you have two parents who are carriers, so both the two parents do not have the disease, but they do carry the abnormal or the defective gene, the red one, right? Which codes for hemoglobin S. And when these two marry, and for each baby that they get, right, you can see that we say you have up to 25% of having a baby with sickle cell disease. You will see this baby, the fourth baby here. So again, this is very important. Sometimes when we show this, people assume that you count 1, 2, 3, 4 babies. Among these, you will find one. No. for every single baby. And this comes from the process of the way the baby is formed. When you have the two pairs of chromosomes coming together from the father and the mother, and you get the zygote and all that, in that process, that's when we say we have 25% chance in each baby that is formed to actually the baby inheriting the defective gene from both the father and the mother. So this means you might find that these parents do not have a single kid with sickle cell disease. Or you might find that these two parents may have all babies with sickle cell disease. That can also happen. Again, if we go back to our book, we have seen that from the hemoglobin gene book and from the sequences or the letters that we read within that book, we all know that when you are reading the letters, you also have neighboring letters. And in sickle cell disease, these are also very important sequence of letters. We call them sickle cell disease haplotypes. And we have different haplotypes depending on the kind of letters that are around the sickle cell mutation. This is again very important to understand because even in Africa, we do have different distribution of the sickle cell disease haplotypes. And these haplotypes lead to sometimes different manifestation of the disease. So when we talk about care for sickle cell disease, when we talk about precision medicine, when we talk about how to handle the different patients, we also need to be aware of the different sickle cell disease haplotype. So for example, the haplotype that is common in East Africa, we call it East Central Africa or Bantu haplotype, which is what we see in Tanzania and DRC and other nearby countries. And it is associated with the most severe disease. Symptoms, I will not go into details of each one of these, but just to show us and appreciate that because, as I showed, red blood cells pass through all the organs. This is a disease that affects all the organs of the body in different ways, So now that we know how sickle cell occurs and how it is inherited, I think we also need to spend some time to look at how we can actually screen for this disease. And again, is what we saw from Dr. Mohammed's talk, because this is a genetic disease, it gives us a leverage to actually screen if we can, even at birth or even before birth. So we can actually do prenatal screening and we can do newborn screening. We can also do adult screening because genes are genes, they don't change, and we are born with them. So we can actually take advantage of this to do early screening because we know even before the baby starts to manifest the disease, we may know way before that this baby carries sickle cell disease, and so we can prepare on how to manage the baby. Even for prenatal screening, the same. So we can also use this to one, help managing the pregnancy better, but also prepare on how we can welcome the sick baby. In other countries, people are also allowed to choose whether they want to terminate the pregnancies or not. And so for newborn screening, this is what we have been doing mostly so this is something that the developed countries have already been able to adopt as national initiatives. And slowly Africa is also coming up. And usually we screen at birth or we screen using the immunization platform. We have different ways of screening and lab methods that we use for diagnosing sickle cell disease. The most conventional ones that have been used for a long time are HPLC, and you can see from this chromatograph that the peak for hemoglobin S. We also have what is known as isoelectric focusing. You can see that you have the different bands and you can distinctly see the band for hemoglobin S. We now have an increasing increasing coming up of point of care test, which is really good. We really want this because we want to increase the access of screening for sickle cell disease, but also the point of care tests that are coming up are very easy to use, even in the very remote areas. And this is what we want so that we are able to screen more and more babies. But we also have molecular and sequencing methods that are mostly used as a definitive methods to establish the genotypes of a patient, whether this person carries the 2 copies of the defected gene, or they are also which haplotypes, or if they also carry other related inherited disorders like beta thalassemia or alpha thalassemia or G6PD and the like, which are really also important when it comes to care for patients with sickle cell disease. Why is it important to do early screening? This is something that we have learned from the developed countries. You will see from this figure in America, they had issues with managing sickle cell disease, but when they introduced newborn screening around 1970, you can see the life expectancy for people with sickle cell disease rose significantly. It's now evident that you can actually prevent and reduce morbidity and mortality for sickle cell disease up to more than 50% if you are able to catch them early. Why is it important to know the genotypes? As I said, one, this helps to manage the disease properly. You will see that if someone has homozygous hemoglobin SS, we say this is a severe disease, or people who have this kind of a genotype will manifest the most severe disease. We also have hemoglobin SC, which is very common in West Africa. And the disease is actually intermediate, not as severe as what we see with people with SS. But we also have co-inheritance, where someone has inherited not only the mutation for sickle cell disease, but also mutations for other related blood disorders like beta thalassemia, where either that would lead to a mild disease or a more severe disease. But this also helps to plan. So because it's a genetic disorder, if a family didn't know that, for example, the picture that I showed, if the family didn't know that they actually carry the sickle mutation because they don't manifest the disease, and then it happens that they got married, they now have a kid with a sickle cell disease, then because this is a disease that is known, is genetic, and we know how it is inherited, we can actually help the family to understand better what to expect and how to prepare for that. So sickle cell disease being a disease that has been here on earth for a very long time, we have had a number of initiatives to develop guidelines for management of sickle cell disease, which covers the continuum of care from birth to adulthood, but also starting with screening and then ensuring the whole point of screening is ensuring that you enroll these babies to comprehensive care. But also we start with prevention. So like breaking the cycle, like what we are doing today, we are giving knowledge so that probably people, before they get married, they can go screen themselves, know their status, know that if you are carrying the sickle mutation, what does that mean? What does that mean to you and to your future family? So that's prevention. But then basic treatment and care, this includes the screening, and then when a child or someone is positive, they're involved, they're enrolled into comprehensive care and support, management of complications that I showed, which are really many. But now we also talk more about the cure. So we do have bone marrow transplant and gene therapy, which bone marrow transplant has in African countries, but now we are coming up. We started talking about gene therapy and it was like a dream, but it's no longer a dream. It's already there. And so this is something that Africa continent is also pursuing because, as we said, sickle cell disease is an African disease. So we do have different guidelines, but this one that I'm showing here is a consolidated guideline from WHO. So I've already spoken a bit. We have, as part of care, we do have the basic care, which we really need to make sure we have. And this is a question that we always get when we talk about the advanced things you do not have. My answer is always like, we cannot afford to always be chasing the wind. We need to do both. We need to now work very hard to ensure that, yes, we do basic time, but here, but also we ensure that we have the advanced therapies as well. Blood donation, this is really important, and it's because we already saw that for people with sickle cell disease, they suffer a lot from anemia, which also leads to different complications. So this is a call to all of us to really participate in blood donation for people with sickle cell disease. This helps in so many ways. It's part of the therapy for stroke prevention, pain crisis management, and the like. But we also know that we need blood from diverse blood donors because of the reasons that Dr. Mohammed already presented. We are different. So also reducing the matching issues and ensuring that we have correct blood that is required for the different people. But we also use this for, now we have a lot of research in terms of transplant using stem cells and regenerative care, which is also something that we can benefit from blood donation. The future of sickle cell disease care, I cannot avoid talking about genomics because I do genomics. So the integration of genomics in sickle cell disease, this is part of the work that we published recently in screening, determination of disease modifiers. We have been working around this because we know the disease, although is caused by a single mutation, but it is manifested differently from one person to another. And we want to understand from the biological point of view, also from the environmental point of view, from social point of view, economical point of view, how all these modify the disease. But also we have seen that we are moving on to precision medicine. And we know that even for other diseases, we don't have one size fits all. You cannot expect to have the same response of a particular therapy, being it a drug or even bone marrow transplant or even gene therapy. We need to understand the different factors that modify response to the different treatments. But also it's really critical for Africa to be participating in translational studies, to be able to sit at the table and negotiate the care that we want to see for patients with sickle cell disease in Africa. And this includes contributing the required data for developing of drugs as well as gene therapies. So just a bit on gene therapy, I think this is very important for us to talk about because this is something that we thought was not possible. It was like a dream, but now it is happening. And we have two different methods of gene therapy. You have ex vivo methods where you remove the cells of a patient, you correct them, and then you bring them back. But we are looking at in vivo method, especially in Africa, because this requires less monitoring. And we hope that if this is done correctly, accurately, targeted well to the sickle mutation or other mutations or variants that are important for sickle cell disease, then you have a more benefit to the patient. So it's important for us to think about this, prepare for this, be involved in the different clinical trials that will be coming to Africa, but be involved while we have proper knowledge of all this and how our data is important to actually develop the best methods for us. We cannot afford to wait and receive therapies that will not work for our patients. Just a quick highlight of different initiatives that we have in Africa for sickle cell disease. This is Sickle in Africa that I'm also part of. It involves 7 countries in Africa, has done research among those in newborn screening in malaria and others. But we also have had Sickle Africa, which was looking at the interaction of malaria and sickle cell disease, again in several countries in Africa. And I think I'll be finishing up. The key messages that we have to think about is early screening. This is really important. Making sure that we have the comprehensive care, the basic care, as well as the advanced care, and we start thinking and keep pushing for precision medicine for sickle cell disease. I thank the patients and families that we work with every day. I have worked very closely with the Sickle Cell Program of Tanzania for more than a decade. Muhimbili University, Tanzania Human Genetics Organization, for which I'm the president, the partners that we work with. Thank you.
Thank you so much for such an in-depth introduction. Please, can we all appreciate Dr. Siana for an in-depth, conversation or introduction, to sickle cell disease. Thank you so much, Dr. Siana. And I think the take-home messages are on the second last slide where you said early screening, comprehensive care, as well as personalized medicine are some of the things that we need to sort of zoom in, especially for the continent. So thank you so much. And also thank you for the work that you are doing as well in this field. And so with that, I'd like to take a few questions for Dr. Siana, if there are any. So if you could raise your hand or type in the chat box, any question you have for her before we move into the panel discussion based on the introduction that she's given. Okay, I think yes. So yes, Dr. Siana, I think you can answer this. What is the relationship between malaria and sickle cell disease?
Yes. Okay, so I think, yes. So I've seen this question several times, and evolutionary, the theory has been malaria was here in Africa, and malaria, the way malaria happens, it affects the red blood cells, right? It's transmitted through red blood cells. And so somehow, as an adaptive measure, people who were genetically born with hemoglobin AS, the carriers, happened to be resistant to malaria. So people are still looking into how the red blood cells of these carriers are different from the red blood cells that we see in people who are normal. Normal is not a good word. People who do not have sickle cell disease, and those red blood cells that we see in people with sickle cell disease. So maybe it is something about the receptors that we find at the surface of the red blood cells, which interact with the malaria parasite. Not just the interaction, but also the hosting of the malaria parasite within the red blood cell, which is different, which made it difficult for the parasite to survive within the red blood cells of people with who are carriers. And so because these are carriers, and as I showed, these are people who do not manifest sickle cell disease. So they're okay. So they keep marrying, and then we have many babies or people who are born with sickle cell disease in Africa. And it's not just in Africa. I think this has also been observed in other parts where malaria was also endemic. And we see also low prevalence of sickle cell disease in areas where they don't have malaria. So for sure, there is an interaction. We are still looking for more answers around this. And with the technology advancement, I'm sure we'll see more and more around this area. Thank you. So I see another question here.
Yes,
How
yes.
often should
Yeah,
HBSS patients get that transfusion, and
yeah.
what are the complications that will be seen due to frequent transfusion? So I'm not sure there's one answer to this. What I know is that as part of comprehensive care, and Arafa is here, Arafa may speak also on this. As part of comprehensive care, usually we encourage people with sickle cell disease to attend routine clinics. And during these clinics, usually they do get thorough checkups. Okay? It is normal for people with sickle cell disease, if you look at their hematological profile, their hemoglobins are low compared to the hemoglobin profile we see in people who do not have sickle cell, even when they are not sick. So around 6.5, 7, and they're okay, they're walking, they're fine. That is like the normal for them. So when, for example, that goes lower than that, of course, they will be anemic, and being anemia is contributed by different factors. We should not forget things like nutrition, which are very basic, what they're eating, right? As part of comprehensive care, they're supposed to be taking things like folic acid. Now we talk about hydroxyurea, all of which lead to better levels of hemoglobin. But if at all there's any factor that leads to them being anemic for whatever reason, they need to be transfused. So we cannot say how many times they should. This will depend on when they are anemic and why they are anemic. But yes, there are disadvantages of frequent transfusions. And one of them is iron overload, which again is another challenge. So even the transfusion itself, the blood components that a patient should be receiving out of the blood need to be very well monitored, ensuring that the patient is receiving what they need. I will say, if Arafa would want to say something about that, please feel free to do that.
Yeah.
Yeah.
Before we invite Rafa, I think most of the questions that are in the chat as well, and thank you, Dr. Siana, for answering that and for giving like an overview of that. Most of the questions I'm told are on the panel, so we can start with the panel questions, and then if there are any more questions that haven't been answered and are on the chat, we can come back to them later on, because
Yeah.
I'm told most of these questions are already on the panel.
Okay. Sounds
Thank
good.
you so much. And actually, that brings me to the first panel conversation, which would be led by Lina. And I would like to hand over to her to introduce the panelists and to steer that conversation. Lina?
Thank you, Ruth. Thank you so much. I'm so glad that we have this panel right after Dr. Siana's presentation. Such a wonderful introduction to sickle cell disease and the information that we need to know. So for this panel, we're going to have Arafa from Tanzania and Sainabu joining us from the UK, but she's originally from the Gambia. So they already did a very wonderful introduction to what they do and who they are. So I would not want to water that down. But just as a reminder, Arafa is, besides being a sickle cell disease warrior, she really does a good job. She is the founder of Sickle Cell Disease Patients Community of Tanzania and does a lot of work around advocacy. And we also have Sainabu Bulaye, who is currently a PhD student at Queen Mary University of London. So Sainabu also does her work around genetics, and she's the founder and chairperson of the Human Genetics Awareness Association. So I would like to invite Arafa and Sainabu to this panel. You're all welcome to unmute. And I think for the sake of time, I will start this off by just asking a very simple question, and I would start with Arafa. So, Arafa, my question to you would be, would you briefly tell us your personal journey with sickle cell disease and how that inspired you to start the community of patients in Tanzania?
Thank you so much, Lina, for this opportunity. As I was introduced, I'm Arafa Salim Said, the founder of Sickle Cell Disease, and I was diagnosed with sickle cell when I was 8 months after birth. Growing up, I saw the misinformation, the misconceptions around sickle cell, and I really used the opportunity to say there's nothing for us without us. Like nobody can walk my shoes than myself. So from there on, I took the initiative of doing advocacy. Before starting the organization, I just was doing advocacy as a person myself living with sickle cell disease until when the time reached and said, you know what, let's take it to another level and do proper advocacy through an organization. So yeah, lack of information, lack of awareness led me to start the sickle cell disease patients community of Tanzania.
Thank you so much, Arafa. And maybe just to ask another question that I had not planned to ask, but since you're doing this work in Tanzania, are you planning on expanding it to the entire region, let's say the East African region, or are you working with other people who are also leading the same kind of groups in other countries?
Definitely, definitely. I'm not doing it only in Dar es Salaam. Like now, I'm in the Western Tanzania, Kigoma, here ready for the World Sickle Cell Day celebration. And yes, I like collaboration. As Dr. Sienna said, I work closely with the Sickle Cell Center in Dar es Salaam. I work with the human genetics. And yes, I work with different organizations, which I helped also start organizations within Tanzania, but also like now I have a role with as a secretary for UMASCO, where it's an entity that brings all African organization that works with sickle cell disease. So yes, I'm not only in Dar es Salaam and Tanzania, I'm actually internationally. Thank you.
Okay, that's amazing. Well done for the good job. And now that you really talk about awareness and you create so much awareness around sickle cell disease, I think it is only fine for me to ask, what is one of the biggest misconceptions about sickle cell disease, in your opinion, that you think the society needs to address?
I think the big the all this comes from is because we don't know how is it how do you get sickle cell. Not many people understand how do a person get sickle cell, because if you look at the parents, both are very okay. Like they will tell you, I never got sick ever until I get my baby. And then now you're telling me my baby has sickle cell. How is that? Because we don't know how the disease can be inherited and how it passed down from parents to parent. So that is one big thing that is really hurting our people. Yeah.
Okay, thank you
Because
so
they
much.
also believe, we also believe, like there's that misconception that if you have sickle cell, you have to look one of the ways that you have to look. You cannot be a normal person. You know, they put a picture behind a person. Yeah.
Yeah, that's so true. So many misconceptions that we need to address as a society. Thank you for sharing those insights. So I will switch to Sainabu before I come back to you, Arafa. So Sainabu, you do your current work on around genetics, and you're also the founder of the Human Genetics Awareness Association. So I would like to know, what is it exactly that inspired you to establish this association, and what is the gap that you're trying to address?
Thank you for your question, Lina. And once again, my name is Sainable, and I am the founder of the Human Genetics Awareness Association. So, starting of the association was because of personal experiences, curiosity, and really wanting to have answers to certain questions that I wanted to know. So, for example, my family is diabetic, and I wanted to answer some of those questions. Why is the family diabetic? Why do some families have hypertension? I also have some family members that do have sickle cell. So I was always wondering, why do some of them have sickle cell and why others are not? So that had always been in my mind as a sort of a general question or terms that I wanted to understand. But I think the one that had most impact on me really was that growing up, I knew like a family member who had a baby that was always isolated and ignored and just left unattended. And I was a part of this. I did not also hold this child because there were certain misconceptions around this child, some certain spiritual stories that was attached to it. But further down the line, I actually studied about genetics in my undergraduate degree, and I later understand that this child might have had a condition called spinal muscular atrophy, which is an inherited condition. And from then on, I felt a sense of responsibility to actually go back and tell people what I've learned or what I know, to find a way that the science that I learned is actually brought to the community that need it the most. So the Human Genetics Awareness Association was actually founded to kind of bridge that gap between the research and the community, but also to kind of lay the foundation for everything that's good about genetics right now. So genetic counseling to genetic testing. So right now, what we're trying to do is just make sure that we cover enough ground that people are aware of what human genetic conditions are, to especially destigmatize most of it, because sickle cell is one of them. But there are many, many other conditions that people need to become aware of and understand. I think one of the main ones that I would think at the top of my head would be albinism. So I think there's a lot of misconceptions that happen around albinism. And I think that's one of the things that we get to talk about and discuss, just so that people know that these are, everyday realities and this is a genetic condition. So that was one of the main reasons behind the formation of the Human Genetics Awareness Association.
I love that. And that will easily lead me to the next question. And I also love that you mentioned that there's this friend of yours who people had different perceptions about just because he had a sickle cell disease. And Arafa also mentioned that people with sickle cell disease are expected to look a certain way, which is a very wrong misconception. So in your opinion, besides just sickle cell disease, there are so many other genetic diseases within our African communities. What is one of the misconceptions that would like you as Sainabu or the Human Genetics Awareness Association you'd like to address around genetic diseases?
I think one of the most common misconceptions about genetic conditions is that it is spiritual. So, because of that belief that most people have, the topic has to be dealt with sensitivity and understanding of people's beliefs and also cultural expectations. But then it's one of the main things. So just get people understand that this is something medical, it is not spiritual. So because most of the time when people think about spiritual conditions, they often associate it with no cure or just accepting as it is and not doing much to kind of manage what the person is going through. So bringing the understanding down to this is medical can actually lead a long way towards just getting people to be integrated into society, because most of the people would be isolated when they do have certain misconceptions around these conditions. But that's one. And the other painful misconception is, for example, if a child is born to, let's say the example that Arafa gave, being born to parents that are usually okay, a lot of stigma falls back on the mother as well. So trying to just get people to understand that, no, this is nobody's fault. It's not the fault of the parent. There should be no one blamed. It's not the fault of the patient. Let us try to come together to really understand how to manage what is happening and, if possible, prevent further cases from happening. And I think that brings me to my third point, which is some conditions are really thought mainly of conditions of the West. that it doesn't apply to us. It is not African. This is not something that used to happen to us. It is because it happened in the West. And I think with more information that we get to know and the more knowledge that we get to share, we are kind of bridging that gap. So that is also one of the reasons why at Huga we established a World Human Genetics Day, and we did that on 30th of March. And we were also privileged to have Mohammed Ansain also speak on that. And what we tried to do there was to break down what is happening in the research field into understandable language for the public. So we invited high school students, we invited policymakers, clinicians, just to get to bridge that gap, just let people understand that genetics is not something to fear, it's something to embrace, and it's something to try to move forward to, as well as make better decisions going forward, if possible.
Thank you for such a detailed response. And I love that there's so much awareness discussion that is going on around sickle cell disease and genetic diseases. So I will now switch back to Arafa. So Arafa, I wanted to ask you a question on the importance of blood donation. And I know Dr. Siana has already given us an overview of this in her presentation. But instead of just asking that directly, I would want you to also address a question that we have on the chat. And someone is asking if there are any complications that would be seen if someone undergoes frequent blood transfusion.
Oh, that question is for me as well.
Yes, you can address that.
Definitely. As a warrior myself, blood is very important. It's more of like it's a life that we mostly depend on. But yes, with frequent blood transfusions, definitely we can get others get iron overload, but also for countries like ours, you can also get what I got a condition that I developed antibodies within my blood. So my blood is fighting with the blood that I get. So I get my HB goes down. So I had to stop transfusions and get treatment. So yes, with frequent blood transfusions, there's a lot of things that could happen. So I think the healthcare workers need to be very careful when we are doing frequent transfusions to patients with sickle cell, because like when you have iron overload, you have the things that happen, and it takes time to be diagnosed what is really happening with you, because it did take a while for me to be diagnosed that I have that condition. So I think we also have to watch out a lot when we're doing transfusions. I know it's more like a treatment, but also have to be very careful. Yeah,
Okay. Thank you, Arafa. So before I come back with my last question to you, I will go back to Sainabu. So Sainabu, I would want to know in a very lay language, what is the importance of genetic testing and genetic counseling? And as you answer that, would you also address a question we have in the chat, which so someone says that couples should get screened before they get married. What happens when they are found to carry the genes? Is there treatment? So I think just to phrase the question in a very simple way, is there treatment for genetic diseases?
Thank you. Thank you for the questions actually. So I think I will start with the first one about the importance of genetic testing. I think the main thing would be understanding what is behind the condition, right? So even before the testing, let's say if you do know that you have, let's say, a child with the condition, I think knowing that it is a genetic condition shifts the dynamics to what you provide and would go to maybe providing care giving support and planning rather than certain other things like blaming or lack of care or calling them certain names or expecting them to have a certain look, like Arafa mentioned. And then again, I think Mohammed and Sienna give a really beautiful introduction and also explanation of the genetics of it, really. I think some people would not know what their status is unless they go and get tested. So I would encourage people to get tested in their various testing areas that they can have in their countries. So having that, knowing your career status, again, now to answer the second question about the marriage, you can't really tell people don't get married or get married and don't have children, right? So people make their own decisions when it comes to these. And I think as an advocate of human genetics, all I can tell you is these are the certain risks that would come with the decision you make. Do you really want to do that? Are you ready for the implications, not only on the affected individual should the child have sickle cell, but also are you ready to provide the care and attention that they would need to have while they're here? So would you say is a cure available for genetic conditions? I think that's a very broad way to put it. But I think Dr. Sienna mentioned like the gene therapies and all of the work that's happening in the sickle cell area, which is very lovely. I wouldn't want to give a blunt yes or no, because I think now there's so much that is happening. There's so much advances that are going on in the field now. So I would just say when you do screen and you know your career status, just ask yourself, is this love more to me than what I'm going to inflict on somebody else when they're born? So you can just ask yourself those questions and hopefully you find the answer to figure that.
Okay. Thank you, Sainabu. So my last question to you would be just as a closing remark. What is one message that you'd like people to know about genetics, about advocacy, and how important it is to our communities?
So for genetics, my one message would be it is not something to fear, but rather it is something to embrace and be enthusiastic about and want to know more of. Because knowing, for example, your predispositions or knowing where your family is or how your family genetics are can let you know or at least try to make certain lifestyle changes. For example, again, using me and myself from being a diabetic family, that would force me to actually at least pay a bit more attention to what I eat and also my activity levels. So that is the first thing. Genetics is something to embrace, be enthusiastic about it. And sorry, what was the other questions that you asked? I've actually lost
How
the
is that
questions.
important to our communities?
Oh, yes. So it is important to our communities because it fosters bond between us, actually. So if you know about human genetic conditions, it allows you to become an advocate in a sense that if you do come across someone spreading inaccurate information or misinformation, you could at least try to not correct them, but at least increase their level of knowledge. So I encourage everyone to become an advocate for it, to raise awareness about it, because although the number of people that are doing human genetics in African populations is increasing, we also rely heavily on the communities to also help us in raising awareness and increasing this genetic literacy that we have. Because when we make informed decisions and when we know about the conditions, we're able to make better choices, we're able to become better people, we're able to become a better community and foster bonds between us. So that's what I have to say. Thank you.
Thank you so much, Sainabu. Now, my last question to Arafa. So, Arafa, what is one message that you'd like people to take away from this conversation on genetic testing and being a sickle cell carrier and general sickle cell disease awareness? What is one message that you'd like people to know?
Hi, thank you so much. I think we'll start first to say to being a sickle cell patient. A sickle cell diagnosis can change our journey, but it does not define our future. So having sickle cell is not the end of the world. Life continues. For the AS, the career patients, like my people, we need to break the cycle. People need to test and make an informed decision early for us to be able to break the sickle cell cycle in the community, in the world. And what was the third one, sorry?
The third one is generally, what would you say is the importance of people creating awareness around sickle cell disease? And what is one message you'd like to leave them with?
Sickle cell is a genetic disease, so please, let's not discriminate, but rather understand and be an advocate, whether for your neighbor, for your kids, for yourself, for the person you love. Yeah, thank you.
Thank you so much, Arafa. Thank you so much, Sainabu. My key takeaways from this is we need to destigmatize all the misconceptions that the society has around genetic diseases. And having sickle cell disease does not define who you are. So you should not be different from other people. And we should embrace talking about genetic diseases. And one statement from Sainabu was, it is medical, it is not spiritual. So thank you so much, Sainabu and Arafa, for joining this panel. And I think if anyone has a question, you can just type them in the chat. If we have time, the panelists will give response to them. Otherwise, they can also just respond in the chat. So thank you so much, Sainabu and Arafa. I will now love to introduce the next panel, which will be moderated by Helen Shikanda. So I will allow Helen to take over from here so she can introduce the panelists for the next panel discussion.
Thank you so much, Lina, for the wonderful moderation. I've been jotting down notes and learning a lot from all the conversations that have been happening here. So for my panel, the panelists don't need introduction because they've already set the stage earlier when our session was starting. That is Dr. Mohammed and Dr. Siana. And they pretty much explained the basics of genetics and sickle cell. And my work is just really to put on the layman shoe and try to ask them questions that maybe your 3-year-old child would ask about sickle cell, or someone who has never been to a genetics class would ask. And I'd start with Dr. Mohammed, who has been explaining to us pretty well about the shelf, and then narrowing down to the book, and then how the instructions in the book could change, and that means a change in our genetics. So I just wanted to ask something about the shape of the sickle cell, like the sickle cell shape that we normally hear. And I remember in our biology class, we were told that it's shaped like a crescent moon.
Yes.
So I'd like to understand now, as someone understands the genetics of and how this changes, what exactly happens when this cell changes like this to our bodies?
Well, thank you so much, Ellen, and to highlight this particular question. So basically, people with sickle cell disease are normally, as I mentioned in my presentation, they are born with a genetic change, meaning that if we remember the book in the library and the instruction manual, there is a change in that instruction, which means now that suddenly that protein that was supposed to be produced by that particular book is no longer working properly. So in this particular context, the sickle cell disease patients, they are born with a genetic change in the hemoglobin gene, as we have heard from Dr. Siana. And this protein, inside these, the protein inside these red blood cells are actually responsible for carrying the oxygen around the body. Now, of course, we have to understand that these red blood cells themselves are not always sickle cell shaped from birth, or we also sometimes call them banana shape. But they have to come under certain conditions, like maybe low oxygen levels, sometimes dehydration, infection, stress, which causes the abnormal hemoglobin to stick together. And then what happens is that they form these long fibers inside the cell. And so then this causes the normal, what would otherwise be normal, flexible, round red blood cell to then become rigid and become sickle shaped or become banana shaped. Now the problem is that these sickle cell cells, they cannot move properly through these small blood vessels. So they become trapped. And when they become trapped, they block the blood flow to the tissues and organs. And of course, as we know, when these tissues that do not receive enough oxygen, then they cause a lot of pain to these patients. And normally, in scientific terminology, we call them vaso-occlusive crisis or crisis. And it is, in fact, one of the most common reasons people with sickle cell disease require hospital care. And as we have also heard from Dr. Siana, that over time, repeated blockages because of these change in the shape will also damage other organs like lungs, kidney, brain, heart. And so it is very important that we understand this properly. Yeah, thanks.
Thank you so much, Dr. Mohammed, for that elaborate explanation. And just to bring you back to the changes that happen and something that Dr. Siana mentioned when she was introducing sickle cell disease about sickle cell being associated with malaria. And we've seen that even in Kenya, most patients with sickle cell disease come from the malaria-prone region. Maybe if you can explain a little bit about these changes in malaria, people in the malaria-prone regions and why most of them are likely to have sickle cell disease. And just to add on that, in case one day malaria will be eliminated, will this also mean that sickle cell will also be on its way to elimination in a way?
Is that question still for me?
Yes.
Okay, yeah. So, I think Dr. Siana already touched on this a little bit, and I just want to sort of expand on what she has already mentioned. So, this is actually a very fascinating example of evolution in humans. Because what is the reality is that, those individuals that inherit one sickle cell gene and one normal gene, we normally call them the trait or carriers. Because remember, again, to remind all of us that for you to really be a patient with sickle cell disease, you have to receive the defective gene from both parents. So both mom and dad will give you the defective gene in the same position, in the same gene, which is when the two pieces of the puzzle come together and then they cause the disease to happen. But what happens sometimes is that you don't receive it from either one of the parents, you only get it from one parent. And so we call that the carrier, meaning you have only one defective gene and one normal gene. Now, it is in this group of people where, they provide or they get protection against severe malaria. Now, again, I think Dr. Siana mentioned this briefly, and this has got to do with the fact that we know that malaria has historically killed millions of children in Africa. And this genetic trait, the carrier status of sickle cell disease, has actually provided some sort of a survival advantage. And that has been passed from one generation to the next. See, the challenge arises when the child will inherit two sickle cell genes, because one from each parent. In that situation, then the protective advantage is then lost, and the individual develops the sickle cell disease. So, in regions where malaria has been common for centuries, the sickle cell gene has become relatively frequent in the population because the carriers of the sickle cell survived malaria better. And as a result, more children were also born with sickle cell disease. What helped protect these populations, the reality is that what really helped these populations from one deadly disease inadvertently created sort of another serious health condition. Your question about what would happen if malaria was eliminated, I think the question here is that what then would happen if sickle cell was eliminated, because it is the sickle trait that offers the malaria protection. And so if sickle cell change was eliminated, would we go back to being more prone to malaria? That is how I would from a scientific point of view ask the question. But yeah, maybe we could get some more highlight also from Dr. Siana.
All right, thank you so much, Dr. Mohammed. I'll come back to you. But now to Dr. Siana, since we are putting this in the most layman way, some people refer to sickle cell as sickle cell disease, but some people also say sickle cell anemia. So in terms of terminologies, what exactly are we supposed to say? Is it sickle cell disease or sickle cell anemia? And what do they mean when you say disease and when you say anemia? Thank you.
Thank you so much. Great question. So if we want to define these terms better, we could say that sickle cell disease is actually a group of diseases which includes the sickle cell anemia. Because with sickle cell disease, we talk about all forms of disease that comes with people inheriting the sickle mutation, right? So the different genotypes. So like we saw people with 2 copies of hemoglobin S inheriting each copy from one parent. We also have hemoglobin H SC, HBSC. So one of the mutation leading to hemoglobin type S and another one hemoglobin type C. So we have people with the genotype hemoglobin SC, which is common in West Africa. But we also have other genotypes. We also have co-inheritance with other conditions like beta thalassemia, which is again some of the changes in the letters around the hemoglobin gene, which sometimes, not always, some of the patients with the sickle mutation also inherit these other mutations. So collectively, we call this sickle cell disease. And then the sickle cell anemia is more specific and usually more related to people who have inherited the 2 copies of hemoglobin S, so the SS. But they are usually used interchangeably. I think the key thing here is to know that when we talk about sickle cell anemia or we talk about sickle cell disease, what we are trying to say is people who have the sickle mutation. I think that's the key issue. Thank you.
All right, thank you so much. That explains it very well. When you were giving your presentation, I think the recommendations of the takeaways that you mentioned was that countries should embrace newborn screening. And in East Africa, I would say Uganda is one of the countries that has embraced that. Like there's a mandatory newborn screening in that country. And in Kenya, I would say maybe Kisumu County, they have started to have that as something that they're taking up. So in your opinion as a geneticist, what would you say about the importance of newborn screening and why countries should take it up as something that they could include in their policies?
Thank you so much. This is actually such an important question. And it's very important because as we saw, newborn screening is an intervention that has been proven to work in developed countries. And it works because we are able to screen and identify the affected individuals early on. So something that we have not mentioned is that And probably Arafa would also want to comment. Arafa is going to beat me because I always go back to her. Apologies, Arafa. But is that the baby who is born with sickle cell disease is born like, a normal, healthy baby for the very first days. And then later on, about, 1.5 years, 2 years, 3 years, that's when they actually start to manifest the disease. And this is also very important to understand because often, even when we have tried to roll out newborn screening and we identified babies who are positive for sickle cell disease, these babies, these families did not come back for results or did not come back for comprehensive care at the beginning because the babies were fine. And then when it gets to 1 year, 2 years, they realize, no, actually something is not right. Then they start to look for care now. Okay. But we know that we are able to screen and identify them early. It is important at different levels. One, at the level of the family, although yes, there will be that denial, but if they happen to understand and even come around and accept that information, even at around that age of 2 years or 1 year, we still have better chances of saving these lives rather than what we often see when we don't have screening in place and we lose more than half of these babies before they get to 5 years. So at family level, it's such an important thing because it helps to identify this baby early on. They get to a comprehensive care, which will eventually reduce the burden of the disease for that particular kid, but also the burden of that disease for the family as a whole. Because taking care of a baby or a patient with sickle cell disease is not easy. So if we are able to identify them early on, enroll them into comprehensive care, we reduce by 50% or more the severity of not the severity, but the complications and the damages that one would experience and even lead to death if they are not properly managed. But if we leave the family, even as a nation, as nations in Africa, knowing that sickle cell disease is a prevalent disease, we need to be able to plan for this group, for these populations within our countries. Always when we go and talk to the ministries of health, they would ask you, how many patients do you have? Like you want them to include a policy, for example, to develop a policy around sickle cell disease, which will talk about these people need, let's say, health insurance coverage, they need hydroxyurea, they need folic acid, they need penicillin, they need this and that. The first question they ask is how many? So newborn screening helps to identify, and what we are doing now is when you identify them, you put them into a registry. They're included in registries, which will show where they're from, right? And slowly we build that database. So for example, in these countries that you're talking about, in Uganda, in Tanzania, I know this because we are part also of the bigger network, the Sickle in Africa network. We have registered up to, I think, about 20,000 babies across the different countries. So if you do that at a country level, at the national level, then you are able, one, to establish the numbers, but you are also able to start knowing the demographics, the distribution, where are these people coming from, which geographical places they are coming from. And this is very critical because this is a genetic disease. There could be different reasons why we see it prevalent in some of the regions and not others. So one of it, yes, is environmental, like we have spoken about malaria. But sometimes it could be lifestyle, consanguinity, for example. So you see that you have many patients that are coming from this region. You ask yourself questions, why? You can't do that. You can't ask yourself such questions if you don't have a registry. You can't have a proper registry if you don't have a proper screening program in place. But also you can now plan, knowing that these are the regions that are more affected. You can plan the interventions around that. So let's say that there is, there are these drugs that are needed. Now you will know, or these services that are needed, these clinics that are needed for sickle cell disease. Now you will know that, okay, we need to make sure in this zone. So for example, in Tanzania, we have different referral hospitals at zones level. So you can know that, okay, I need to make sure in these regional hospitals or these zonal hospitals, sickle cell disease management is well integrated because we have a number of patients that are coming from this place. Not that you won't have it in other places, but you need now to concentrate and distribute your resources accordingly. You cannot do that properly if you don't have the numbers, if you don't know the distribution, if you don't know where these people are coming from. Yeah. So we have benefits of early screening in different ways and at different levels, at family levels, at community level, up to the national level. Thank you.
Thank you so much, Dr. Siana, for that explanation. You had mentioned Arafa. I don't know if you, Arafa, you want to respond to that as well.
Yes, sure. Thank you so much. As Dr. Siana said, we have a program on newborn screening, and when they're only like when they're newly diagnosed, we do see that because they start treatment early, so even getting a lot of complications is not easy. Like you can get complications, but they're not that big. So early screening does really help indeed. And I am part of awareness part on the newborn screening for the parents and all that. And yeah, I really applaud the work that has been done, because we all know how the complications can really affect people living with sickle cell disease. With early diagnosis does really help, because most of the patients that we see are late diagnosed and with a lot of complications. Yeah, thank you so much.
Thank you for that. My next question is to both Dr. Mohammed and Dr. Siana, whoever will respond to first. And it's generally about the advancements that we have in sickle cell treatment and management. And Dr. Siana talked a bit about the genetic CRISPR gene editing, stem cell, and bone marrow transplant, and that in Africa, most of the time, we've seen that we've started having bone marrow transplant as a form of treatment and management. So when we talk about these advancements such as CRISPR gene editing, which are too expensive and generally out of reach for African patients, is there any alternative that we can have to that? Or do we see, when are we likely to have these technologies in Africa so that we can have our patients with sickle cell also get that kind of treatment?
Okay, thank you so much, Ellen, for this important question. And I think we are living in a very exciting time where there have been some significant advancements in the sickle cell management. And I think you rightfully mentioned that, scientists have come up with things like CRISPR, which is a gene editing tool. And normally people have a tough time understanding what is CRISPR. I always try and explain it in a way that think of it as a scissor, but at the size of a molecule. So it's a scissor that goes and sits on the DNA where that change, that letter that we saw from Dr. Siana's slide, where that scissor goes and it cuts off that position where the letter has changed. And that's the very basic analogy of how CRISPR works. But actually, CRISPR gene editing is, in fact, one of the most exciting developments in modern medicine, because it really allows scientists to make very precise change to that DNA. And normally in sickle cell disease, what would happen is that the doctors would first collect these blood-forming stem cells from the patient. So all the cells that you know would eventually, as they mature, will actually form that red blood cell. And then what happens is that they modify those cells in the lab. And what they're modifying is actually the DNA that sits inside that cell. So they take the entire library, and then from the library, they look for that specific shelf. In that shelf, they look for that specific book. And in that book, they look for that specific chapter, and then they remove it. And then what they then do is then they return these cells back to the patient. So then what happens is that these modified cells or edited cells, they can then produce healthy red blood cells, which then will not sickle. Now, some of these therapies, of course, have been already approved in parts of the world, particularly in high-income countries and the Western world. And what we are seeing, and the evidence is showing that there are some remarkable results. Many patients are beginning to become free from these painful crises. But of course, one of the biggest issues right now is that these treatments are currently very expensive. So we're looking at almost $2 million per patient, which is extremely expensive, not only for an African, but even for somebody who's staying in the US. It's very expensive. And they require very highly specialized medical facilities and expertise. So the question is not just, do we have CRISPR? It's about, can we deliver CRISPR to the patient and then be able to monitor that patient after delivering the treatment? So that means that it is not about the science, it's also about the infrastructure. It's also about that clinician knowing, having the skill set to know how to manage the patient, even post the therapy, how to monitor the patient. Now, at the moment, there is no gene editing facility or trial that is currently happening in Africa, but I sincerely believe that this is rapidly going to change. And I think that Africa will eventually benefit from these technologies, because the African scientists are really shouting at the top of their voices to make this a reality in our continent. But again, we also have to be pragmatic. We have to be practical. The access, and not just access, but we want widespread access, because it's not about one country, it's about 54 countries. We want this access to all of them. And to enable that, we will really have to get a proper investment in the infrastructure that is required, in the laboratory capacity that is needed, in training of healthcare professionals, in putting in the regulatory system, and then the sustainability plan in terms of the financing mechanism. So all of that has to really come in properly as an ecosystem that will then become a process where patients with sickle cell can actually get the curative treatment option. But of course, in the meantime, we should also not forget that gene therapy is not the only way, and that even today we can save lives through newborn screening, as we've heard from Dr. Siana mentioning the importance of newborn screening and how it really informs the government. So that is very, very important. We should also talk about the importance of having access to drugs like hydroxyurea, which is very important in the delaying of the maturation of fetal hemoglobin to adult hemoglobin. Then you should also have an understanding on the importance of vaccination to children with sickle cell, and comprehensive care. And I think we've also heard from both Sainabu and Rafa about the patient education. So a lot of these activities will really help us understand the full picture and not only run towards the editing and gene therapy, which is at the moment very expensive, but we should also not forget our basics, which is prevention. We should not forget our basics, which is the hydroxyurea and the newborn screenings and the education. So while we are working towards tomorrow's cures, we also must continue to improve today's care. That's what I would say.
Thank you so much, Dr. Mohammed, for that positive response. Dr. Siana, do you have something to add in that line? The same question that I've asked about whether the new advancements are within reach in Africa.
Yeah, so thank you so much for that question. Again, an important one. So as I said before, we always, when we talk about advanced treatments for sickle cell disease in Africa, we always get this like a back clash, something like you're talking about bone marrow transplant, you're talking about gene therapy, you're talking about all these advanced research that you want to do, and yet you don't even have universal screening programs for like newborn screening. And my opinion always is that in as much as we recognize that we need to have the basic care in place, but if you ask me, I would say when America was establishing newborn screening, that was 1970, right? That was way after sickle cell disease was discovered. We already knew that this is a disease that is prevalent in Africa, because even the screening that they were doing in America was sort of more tailored to African Americans, because they could see that these are the most affected population. The question is, when they were starting newborn screening in 1970, where was Africa? And how many years now? And we are still talking about newborn screening. So if we are not careful, we will again chase the wind. We will find ourselves in the same situation when they will be talking about gene therapy or bone marrow transplant or small molecules development, alternative drugs for sickle cell disease. For developed countries, and we are only focusing on the basic care. So we'll find ourselves in a very similar position as what is happening with the newborn screening now. And we don't want that. And the only way to break that is to do both. Strengthen the basic care, but at the same time, start working very hard to ensure that we are at the forefront of development of alternative therapies. And I agree with Dr. Mohammed, this is not just gene therapy. There's still a lot of research undergoing now on repurposing different drugs for different diseases. This could also be one of the approaches. Identifying different small molecules that can be used for therapies. And we are already seeing a number of molecules in the pipeline for sickle cell disease. But the challenge is the same. We are on the receiving end. And even being in the receiving end, we are not even participating fully in the clinical trials, even for drugs, let alone gene therapy. But we are also seeing that companies are now coming to Africa, companies that are developing gene therapies for sickle cell disease. They are now coming to Africa to see how they can involve the African populations in this process. So what I'm trying to say is we do both. We have to make sure we strengthen the basic care, but at the same time, we are investing as much as we can in the advanced care, because if we talk about sickle cell disease we talk about Africa. We don't have an option. We have to do that. We have an obligation to ensure that Africa is not left behind in this move. And we do not want to be on the receiving end, because we already know from our genetic backgrounds the diversity that we have. We might have drugs or therapies that will not work in Africa if they are not developed in consideration to how our genes are working. I am not hesitant that this advanced therapy will come. Even if we talk about bone marrow transplant, just a few years ago, this was something seemingly impossible, but now it is no longer the case. And we are already seeing that these are things that can be done in Africa. We just need to sit and analyze, yes, the cost is high. Why is the cost high? Usually it's because of the things that are required, the different things that are required for actually establishing. And then start to break this down into milestones or different ways that we can actually make sure that we are developing or we are manufacturing some of these things in Africa, some of the things that we know that will be required for these different. So we start to position ourselves. I know, for example, in Uganda, they have been funded to develop, to be part of developing gene therapy for HIV. So this already shows us that this is coming. It's no longer a dream, it is coming. It could be 2 years, 5 years, 10 years, but it will come. The question is, when it comes, how are we positioned to ensure, first of all, the safety of that in Africa? Thank you.
Thank you for that, Lina. And I know the scientists in the room here will ensure that Africans will not just chase the wins, but actively participate. And that reminds me of an annex in the pandemic agreement that is a bit contentious regarding the pathogen access benefit sharing, where the global north wants to get the pathogens from Africa, let's say from the Ebola virus, and come up with vaccines, but we have nothing to benefit from it. Same way you're talking about pharmaceutical companies coming to Africa to sort of sell the drugs here, but we are not getting anything from it. So because time is almost running out, then we will just have a last round of comments from you. If there's one thing that you would like people to know about sickle cell, especially with so many misconceptions that we have around sickle cell, what would you want people to know? I'll start with you, Dr. Mohammed.
Okay, thank you so much, Ellen, and really great conversation, actually. For me, if I could leave people with one message, it would be that sickle cell disease is manageable, and people living with it can actually lead a very long, productive, and fulfilling lives when they receive the timely diagnosis, when they get appropriate care, and when there is support from their families and communities. You know, many times we only focus on only on the disease and not on the person. But children with sickle cell disease can go to school, adults can have a career, they can have a family, and of course, even these patients have a lot to contribute to the society. And what really makes the biggest difference is not only medicine, but it's also the understanding, the compassion, and the access to quality healthcare. So for me, sickle cell disease should really not define a person's future, because with the right support, individuals living with sickle cell disease can actually thrive. Yeah, thank you.
Thank you so much for that. And you remind me of a story that I did last year of sickle cell patients who are over 65 years old. So it's possible to live beyond what people usually say, that when they reach a certain age, then they'll not survive. So thank you for that. Dr. Siane, your final comments and something you'd like people to have in mind when they think about sickle cell. Thank you.
Thank you so much. So I think for me, I see here we are 31 participants. And as you say, we have people coming from different disciplines, working in different things. I think my final comment would be, and the fact that we are all here, it means we care about sickle cell disease and what we want to see in Africa. And so my final comment would be just to ask ourselves, each one of us, how we want to contribute towards that. Thank you.
Thank you so much, Dr. Siana and Dr. Mohammed, for the wonderful conversations that we've had and for breaking it down for people who do not understand genetics and the science behind sickle cell disease. So I'll give it back to Ruth for the second part of the program. Thank you.
Thank you so much, Helen, for an excellent moderation, and thank you to our panelists, Mohammed and Siana. Thank you so much for enlightening us. I think there's just 3 quick questions that we can do. I am aware we are running a bit behind time. Just questions that were already on the chat and were not previously answered that I would invite our panelists to answer. I think I would start with the one, the most recent one, which would relate to what Sian and Mohammed spoke about. And maybe either of you could answer this. What method should we adapt to make individuals and families aware of sickle cell disease and to get carrier screening done? Mohammed?
Okay, I thought Siana was going to answer that. I think for me, what is really important as far as methods are concerned to these families is, awareness and education. So the whole concept of advocacy is extremely important. We've heard the story of Sainabu, and I think that story also resonates with the Tanzania Human Genetics Organization when we started 7 years ago. It was through advocacy that we actually got people to first understand what is genetics, to really be able to then understand why is genetics so important and what are some of the problems associated with these genetic conditions. So really, I think awareness can only be done, through public education, advocacy, and I would not limit ourselves to just the patients or family caregivers. I think the awareness has to be mass awareness, meaning if you have an opportunity to engage with the media, and I'm so glad to see that Helen is part of this panel, because this is where we really need media people to be part of these kinds of conversations, because they will help to amplify. So if you have an opportunity to speak to the radio, to the television, to the newspaper, to your social media, uh, you know, very recently we even started producing things on TikTok, which I didn't know anything about. But I realized that that is also so important because there are consumers of that particular platform. So whatever medium you want to use it to amplify that voice so that you are reaching out to the most important people. And the second group of people you should also be targeting are the policymakers, because eventually all decisions around getting the infrastructure, getting the funding to do the carrier screening and all of those things will actually come from our policymakers. So mass education and policy advocacy is very, very important.
Thank you so much, Mohammed. And quickly coming back to you, Siana, if you could respond to the question about mitochondrial dysfunction. So Usman asks, could mitochondrial dysfunction predict sickle cell disease and organ damage independently of traditional biomarkers? And can mitochondrial DNA enable real-time crisis risk assessment?
Okay, so I don't know. I've never done that. Could be. I would love to hear more about why we thought of mitochondria, because we all know from biology that mitochondrial DNA comes mostly from the mother, right? So why would we want to approach it from the maternal side, while we know that this is a disease that is genetically inherited, one from somatic genes, chromosomes, and not mitochondria, and both from the father and the mother. So
Okay.
maybe if we would like to look at it from the angle of disease modifiers, yes, that would work, especially because of the fact that we know there's a lot of relationship in terms of energy production for the red blood cells, and how that also influenced the disease in sickle cell disease that we have seen. We have also seen associations with some of the genetic markers that are actually related to the energy cycle within the red blood cells in sickle cell disease. So if you talk about that, I think yes. But in terms of inheritance or predicting the disease itself, in general, I think this is something that we can explore. I can't say no, because this is science. Science is very broad. Science is not limited. So anything is possible. Thank you.
Thank you. Can I
Ruth, can
invite
I?
Mohammed?
Yeah, I just wanted to also sort of piggyback on Sienna's answer, and I think this question is quite important. The thing is that this is a very plausible hypothesis, okay? But again, I would be very careful because it really requires proper clinical validation. You see, in sickle cell disease, there are traditional biomarkers. For example, you've got hemoglobin, you've got LDH, you've got bilirubin, you've got reticulocyte count, you've got HbF, white blood cell count, CRP. All these are very useful parameters, but they don't really fully explain why some patients would develop severe organ damage while others do not. Now, you have to first ask yourself, what is it that mitochondrial dysfunction can capture as a different biological layer? you know so if we understand our biology about mitochondria, it would give you information about oxidative stress, it will give you information about inflammation, it will give you information about tissue damage, or even immune activation. So the cell-free mitochondrial DNA is interesting because it can technically, what they claim is that it can work as a biomarker, but it can also be a mediator of inflammation. And if it releases in the circulation, um, it can actually behave like a damage associated molecular pattern. Okay. And it can maybe help us understand a bit what is happening in terms of the stress related to the vasoacculation, meaning the pain crisis. So measuring it can help identify patients which have a high risk of crisis or a high risk of organ injury. But having said that, I would be very cautious not to overstate it. Okay. Because again, we have to see this right now as an emerging biomarker, and it is not yet a validated sort of clinical test. So the research question is whether it can predict the crisis or end organ damage independently of what we already are using as established marker. And to do that, we really will require longitudinal studies, with testing all these parameters properly. Yeah. And maybe one more
Yeah.
thing to add. There was a question that was very interesting, and I don't know if we had a chance to
Yeah.
answer. Particularly which touched on the importance of phytomedicines,
Yeah.
right? And this is actually very interesting because it's a very fascinating field that most of these clinically studied phytomedicines have actually come from West Africa, particularly from Nigeria. Okay, the important thing is, however, that whenever we are talking of phytomedicines, we first have to very much understand that they have to go through a proper way of clinical validation. All right, It's a very promising area of research, especially in Africa, but they should be viewed as adjunctive therapies rather than replacements. Okay. So I'll be very careful of saying that, oh, let's use a phytomedicine to replace what we are now using as hydroxyurea or blood transfusion or infection. No, we should not be looking at that. And to just sort of give you an example, that there are medicines like there is Nicosan, which is found from Nigeria. It was developed in Nigeria as a traditional medicine, and then later researchers, within the Nigerian framework, actually said that this might be have an influence on the vasoocclusive crisis. So it actually reduces the pain. So it's currently under phase 2 clinical trial. Okay. And then there is also another drug called Cyclavit, which is, in fact, comes from pigeon pea. And, this is also another sort of, phytomedicine where, it reduces these painful crises. So again, there are lots of opportunities, lots of important avenues to look at phytomedicines, but I would not replace them with current ones. And I would also investigate it from a very scientific approach, utilizing our regulatory bodies before we even claim that they work. Yeah.
Thank you, Mohammed, for clarifying that. And just to follow up on that discussion of phytomedicines, I think there was a question as well around the role of nutrition as well. So if either of you could also clarify on the role of nutrition in managing the disease, would appreciate that. Sienna, please.
Sorry, Rose, I had to pick up a call. I'm sorry.
Oh, okay, no worries. Yeah.
Did you want me to say something?
Oh, yeah, to respond on the role of nutrition in the management of the disease. Yeah.
All right. Yeah. So that's a good one and a very basic question, which I think, again, when we talk about basic knowledge and care for sickle cell disease, this is really important. If you talk to Arafa, she will tell you how she manages herself in terms of water intake, in terms of balanced diet that she has to ensure she takes and all that. And this is important. And sometimes when we talk about balanced diet in Africa, people are thinking of chicken, fish, and all that, but not always the case. Even the simple meals, yeah, making sure that you have your, I don't know if in other places they have ugali, but for Tanzania we have ugali and vegetables and beans and, sardines and things like those. Ensuring that you are getting all the nutrients is critical. If you can get foods that, of course, will also help you to boost the hemoglobin, like some of the vegetables that we know them, even from our families in our communities. It's good to do that. And then proper hydration, because we already know the effects of dehydration for the red blood cells and all that. I don't want to go so much into the role of microbiome as well in sickle cell disease, but of course we know microbiome profiles that we have is also influenced so much by what we eat, but we also know that the microbiome, especially the gut microbiome that we have, also is associated with a lot of things, including metabolism of different things, including the drugs. So again, looking at nutrition is quite a broad thing. But just to say that paying attention to having balanced diet and a lot of water. And I'm also very passionate about traditional medicine research in the sense of identifying some of the plants that we have that are medicinal in our populations. I think China, India, I think they have done well in this area, and we have not done so well in Africa. And yet we do have these plants. So I think if we have people in the group who are coming from this discipline. Please work on this. I'm pretty sure we have a lot of plants that which are medicinal, that we can study scientifically, prove the kind of chemicals or molecules that are in these plants that we can develop drugs from. And it's important to do this rather than just waiting for drugs that are coming from elsewhere. Thank you.
Thank you so much, Siana. And so with that brings us to the end of the panel conversations and the keynote as well. I hope we've been able to answer all the questions. I hope there isn't a question I skipped. If you feel like I skipped your question, please just retype it in the chat box and we can come back to it. But at this juncture, I'd like to invite all of us to share our cameras so we can take the group photo quickly. Before we move into the next session. So if you could quickly turn on your camera. And while turning on your camera, let's appreciate our panelists, Dr. Mohammed, Dr. Siana, Arafa, Sainabu, for an excellent job in breaking down the sickle cell disease and genetic conditions in general. So thank you guys. We really appreciate that. And hopefully all of us can be able to continue amplifying the work that you guys are already doing, in our own small way, small capacity, So thank you so much for, blessing us with your time and with your knowledge.